So content of my presentation is introduction, the key concepts and definitions, the types of mutations, importance of genetic toxicology assessment, case study, and genetic toxicology assays. So first, what is genetic toxicology? So first term is a genetic, which refers to DNA and chromosomes. Genetic refers to the impact of toxic substances on organisms' genetic material, particularly the DNA and the chromosomes. And the second term is toxicology, which refers to the study of harmful effects that substances such as chemicals or the radiation or other environmental agents. We have to study those effects on biological systems, and particularly we have to focus on the genetic material such as DNA and the chromosomes again. So first we should understand the term, what is gene and what is chromosome. So in a simple language, the chromosome is a thread like structure which is located inside the nucleus of animal or plant cells. So each chromosome is made up of protein and a DNA. So you can see in the diagram, there is a cell, inside there is a nucleus, and there are chromosomes. So in another language, we can say that the chromosomes are a long, tightly coiled strands of condensed DNA and proteins. And what is DNA? So DNA is a long, continuous strand of genetic information. So what is gene? Gene are a segment of DNA which controls the inheritance of specific characteristics or the traits. So in another diagram, you can see there are two genes, gene A and gene B. What is gene A? Here gene A is the DNA sequence which produces a protein A, and the gene B is the DNA sequence which produces the protein B. So if there is a mutation in the gene, it failed to produce the specific protein. Maybe if there is a mutation in gene A, it failed to produce protein A, or it failed to produce protein B. So in a simple language, layman language, the DNA is like a long piece of string. The genes are the knots in the strings, and the chromosomes are like a big coil of the knots. So now there are two major concepts, what is DNA and what is RNA. In simple language, DNA is a double stranded material and RNA is a single stranded. So in DNA, it contains a deoxyribose as the sugar, that is H, and in RNA there is a ribose sugar, which is OH. There is a slight difference in the bases used. In DNA, there is a thymine, cytosine, adenine, and guanine. And in the RNA, instead of thymine, there is a uracil, that is U. So now moving toward the genetic toxicology. Genetic toxicology is the study of the genetic damage to the hereditary material that results in the genetic alterations, which includes the mutagenicity, transmissible genetic alterations, and the genotoxicity. So, in simple language, it is the study of how chemicals or how radiation or how environmental factors can cause a genetic damage in the living organisms. So there are key aspects in the genetic toxicology. First key aspect is DNA repair, DNA damage. The second key aspect is mutagenicity testing. Then third is the carcinogenicity assessment, and then regulatory guidelines for the genetic toxicology assessments. So now I am going to cover the key concepts and the definition in the genetic toxicology. So first part is what is mutagen, what is mutagenic, and what is mutagenicity. So mutagen is a physical, chemical, or biological agent that cause the mutation. It causes the permanent changes in the DNA sequence of an organism, which leads to the genetic disorders or maybe cancer or maybe any cellular dysfunctions. So there are different examples of mutagens. So first example is a physical mutagen, mainly radiations. So it includes the UV rays, the X rays, the gamma rays, etc. Then the second is a chemical mutagens, which we are routinely dealing with, the agrochemicals mainly, or the alkylating agents, the benzene, nitrosamines. Those are chemical mutagens. And the third one is a biological mutagen, so which include certain viruses like human papilloma virus. So this virus disturb the normal DNA function, so which is part of a biological mutagen. So the term mutagenic, which refers to the ability of substance to induce the mutation, and the term mutagenicity, which refers to ability of substance to cause the mutation, that is permanent changes in the DNA sequence of an organism. Then second term is a genotoxicity. So genotoxicity refers to the ability of substance or agent that cause damage in the genetic material. So which leads to the mutations or the chromosomal aberrations or the cancer. So, genotoxicity is a broader concept which encompasses the mutagenicity, the clastogenicity, the anogenicity, so which includes the various types of genetic damage, not only the mutations. So, there are key differences between the genotoxicity and the mutagenicity. So, genotoxicity is a broad term for DNA, RNA, and chromosomal damage, and mutagenicity is specific to mutations, that is permanent changes in the DNA sequence. In the genotoxicity, which includes the mutagenicity, clastogenicity, and the anogenicity, and in the mutagenicity, which is a subset of the genotoxicity and which focuses only on the mutation induction. Then the effects. So, genotoxicity can cause DNA strand breaks. It can cause chromosomal aberrations, oxidative stress, or aneuploidy. The mutagenicity lead to point mutations, the gene deletions, translocations, or replication errors. Then the examples of genotoxicity are ionizing radiations, the oxidative stress, the alkylating agents. Mutagenicity examples quite similar. So those are UV radiation, nitrosamines, X-rays, benzene, etc. For testing genotoxicity assessment, the testing methods are comet-assay, chromosomal aberration, or micronucleus test. And for mutation, to detect the point mutation, basically, you can conduct the bacterial reverse mutation assay or a cell gene mutation test. The next terminology is carcinogen, the carcinogenic, and carcinogenicity. So first, carcinogen. So carcinogen means cancer-causing substances or the substance or agent that can cause a cancer by inducing the genetic mutation or by disturbing the cellular processes. So the examples of carcinogen are tobacco or tobacco smoke, then certain chemicals, and again the UV radiation may cause the cancer. The carcinogenic is a property of that substance to cause the cancer. So example is a UV radiation is a carcinogenic because it can damage a DNA which lead to a skin cancer. And the carcinogenicity means the ability or tendency of that substance to cause the cancer. So it often assessed by experimental studies. The example of carcinogenicity is a benzene. It is well documented basically. So what is the difference between the carcinogen and the mutagen? So carcinogen, which promotes the cancer formation, and the mutagen is a substance that cause a genetic mutation in the DNA. So carcinogen, which leads to uncontrolled cell growth, it causes tumor formation and finally the cancer. And the mutagen, it causes the changes in the DNA sequence leading to genetic disorders or maybe cellular dysfunctions. So in the carcinogen, it often disturbs the cell cycle regulation, so which damages the tumor suppressor genes. And in mutagen, it alters the nucleotide sequence. So it induces the chromosomal abnormalities, which interferes with the DNA replication. So I have already explained the examples, but there is a slight overlap between the carcinogen and mutagen concepts. So some carcinogens are also mutagens because they induce damage, and some mutagens can lead to carcinogenesis if they cause mutations in oncogenes, cancer causing genes, or tumor suppressor genes. The next most important concept is a clastogen, the clastogenic and the clastogenicity. So clastogens means a substance or agent that cause a structural chromosomal aberrations. So it may be a chromosome break, chromosome gap, deletion, eccentric fragments, translocation, rearrangement, etc. So examples are ionizing radiations, again the chemicals like ethylene oxide, benzene, etc. The next is a clastogenic, means property of that substance to cause the structural chromosomal aberrations. So example is a X-ray. So X-ray are clastogenic because they induce the chromosome fragmentation. The next term is a clastogenicity. So it's the ability or tendency of agent to cause the chromosomal damage, often assessed using a cytogenetic tests. So you can conduct in vitro chromosomal aberration or in vitro micronucleus test to detect the clastogenic potential of the test item. So example is a clastogenicity of ethylene oxide, which is evaluated through micronucleus assay or chromosomal aberration tests. The next term is a anugenic, and anugenicity. So what is anugen? So anugen is a substance that cause abnormal misaggregation, so which leads to aneuploidy, means there is a abnormal chromosome number in the cells. For example, a colchicine or vinblastine, those are known anugens. Then anugenic, the property of that substance to cause the aneuploidy. So again, the example vinblastine or vincristine is anugenic because it disturbs the microtubule dynamics and it causes the chromosome misaggregation. Then anugenicity. So it's the ability or tendency of an agent to cause the aneuploidy, that is abnormal chromosome number in the cell, and which is again assessed by the cytogenetic tests. So micronucleus test and fluorescence in situ hybridization are those two techniques which are used to determine the anugenic potential of the test item. So here I have given the example of colchicine. Then the next most important terminologies are somatic cell and the germ cell. So somatic cells are the cells in any cell in the body except the germ cells, means sperm and the egg. So somatic cells can make up the tissues or organs like skin, liver, brain, muscle. Like germ cells, the mutations in the somatic cells are not inherited, and it affect only that particular individual, not the offspring. The germ cell means a reproductive cell that participates in the sexual reproduction, giving rise to sperm or the egg cells. But it passes the genetic information to the offspring. Then types of mutations. So there are four different types of mutation. First is based on cell type. Second one is based on the structural changes. Then third one is based on the functional consequences, and the fourth one is based on the origin. So first is based on the cell types, that is somatic and germ cell. So what is somatic and germ cell mutation? Somatic cell mutation occur in a somatic cells such as skin, liver, or lung cells. And the germ cell mutation occurs in germ cells that are involved in the reproduction, as I said earlier, sperm or egg. So somatic cell mutation is not inherited, and it affects only the individual, but germ cell pass to the offspring and it affect the future generations. So effect of somatic cell mutations is that can cause cancer or premature aging or localized tissue defects due to the mutation. And germ cell mutation can cause a genetic disorders like cystic fibrosis or sickle cell anemia. So examples, skin cancer due to UV radiation or lung cancer due to smoking. So those are restricted to that individual. So those are somatic cell mutations. And Down syndrome or hemophilia, those are passed to the offspring from the parent. So these are germ cell mutation. Then somatic cell mutation can be acquired due to environmental damage, that is radiation or the chemical. But for the germ cell mutation, it can be spontaneous or mostly those are inherited only from the parents. So this is classic example of somatic and germ-line mutation. So it gives a local effect for somatic mutation, and germ-line mutation, the entire organism will be affected. Then the second is mutations. Those are based on the structural changes. These are first is a point mutation. So point mutation means changes in the single nucleotide. So there are three subtypes in the point mutations. First is the substitution. For example, A to G substitution. Then the insertion means extra nucleotide is added, and the deletion, the nucleotide is removed. The next one is a frame shift mutation. So it is caused by the insertions or deletions which shifts the entire frame so that there is a alteration in the protein synthesis due to shifting of the frame. The next is a chromosomal mutations. So the types are a deletion. So there is a loss of chromosome segment or the duplication. The extra copies of the segments are added, or the translocation. So chromosome segment swap between the non-homologous chromosomes, or the inversions. A segment flip within the same chromosome. So these are the types of the mutation. So first, you can see in the diagram, there is a deletion, last green colored chromosome. Then there is a duplication, again last two green chromosomes, you can see. Then there is inversion, there is insertion, then there is a translocation. You can see the green and yellow part, and numerical as well, the polyploidy or the aneuploidy, that is abnormal chromosome number. So again, the mutations based on the functional consequences. So there are again three types. First is a silent mutation, then the missense mutation, and then nonsense mutation. So what is silence mutation? So no change in the protein structure or function due to the mutation. The second one is a missense mutation. So missense mutation, which alters one amino acid in the protein, which affect the function. So classic example is sickle cell anemia. So sickle cell anemia is caused by the missense mutation in beta globin gene at chromosome number 11. So which results in a single nucleotide substitution and which alters the amino acid sequence. So normal codon is GAG, which codes for the glutamic acid. And if there is a mutated codon, so instead of GAG, if there is a GTC, then it codes for the valine. So it change from hydrophilic amino acid to hydrophobic amino acid, which leads to the abnormal hemoglobin aggregation, and the cells become the sickle shaped. So this is a missense mutation. So first is a silent mutation. There is a change in the nucleotide. You can see in the first part of the figure. So there is a change in the nucleotide A to G, but there is no change in the amino acid. Then the next is a missense mutation. So there is a change in the amino acid. Sorry, the change in the nucleotide A to C, and there is a change in the protein synthesis. The third one is a nonsense mutation. So in the nonsense mutation, it converts a normal codon into the stop codon. So there is a premature protein termination due to the incorporation of the stop codon. The classic example is a cystic fibrosis. So cystic fibrosis is a genetic disease which is caused by the CFTR gene, cystic fibrosis transmembrane conductance regulator. So in a normal, when there is a normal codon, then the codon is GGA, that is glycine, and if there is a mutated codon, then the codon is the sequence is TGA. So there is an addition of stop codon basically, so which produce the truncated non-functional CFTR protein. So you can see here there is a GAA, and now there is a nucleotide change. There is addition of stop codon, that is UAA. The next is a mutation based on the origin. So first is a spontaneous mutation, and second is a induced mutation. So spontaneous mutation occurs naturally due to the DNA replication errors, and which is not influenced by the external factors. This which is spontaneous basically. And the induced mutations, those are caused by the environmental factors such as radiations or chemicals. So examples of induced mutations are UV radiation, which cause the thymidine dimer in the DNA. So in the first figure, you can see there is an induced mutation because of UV light, radiation, or maybe chemical, and the spontaneous mutation. So maybe error in the DNA repair, the error in the transcription, or error in the polymerization, or error in the replication. So why there is a need for genetic toxicology assessment? So we saw the different types of mutations and what is gene, what is chromosome. So we should now understand what the importance of the genetic toxicology assessment. So first is to identify mutagenic and carcinogenic risks. Genetic toxicology assessment, it helps to detect the changes, detect the substances that can cause the mutations, so to prevent the genetic disorders and the cancer. It also provides the insights that how chemical agents or radiation or pollutants that impact the genetic stability. The second is ensuring drug and chemical safety. So regulatory bodies like FDA, EMA, OECD, ICH, those require genetic toxicity testing for the drug approval. So which evaluates the new pharmaceuticals as formulations or agrochemicals or other uh, and pose the genetic risk. And it also prevents the release of mutagenic compound into the market. Third is environmental and occupational hazard assessment. So it monitors the DNA damage caused by the industrial pollutants, the pesticides, and the heavy metals. So it ensures the safety of workers as well, which are exposed to radiation, toxic fumes, or the carcinogens. It also helps to formulate environmental policies to reduce the genetic risks. The fourth one is understanding mechanisms of cancer development. So it link the genetic mutations and the tumor formation, and it also helps in the development of targeted cancer therapies, or it helps in the precision medicine basically. Then the advancing biotechnology, so it helps to refine the genetic modification techniques to prevent the unintended DNA alterations. So overall, the genetic toxicology plays a very crucial role in the scientific advancements, in the regulatory decision making, and protecting the human health from the genetic damage. So there are toxicology assays. So there are few battery assays of genotoxicity. The first one is a bacterial reverse mutation assay or AIMS test, which is developed by the Bruce Ames. The second assay is in vitro chromosomal aberration assay, or you can conduct in vitro micronucleus test. And the third is a in vivo micronucleus test, or you can conduct in vivo chromosomal aberration test. The fourth genotoxicity assay is a cell gene mutation assay, then comet assay, sister chromatid exchange assay, transgenic assay, or there are in silico approaches like use of Vega software for in silico detection of genotoxicity impurities is also available. So for today, I'm going to I'm going to teach about only the bacterial reverse mutation assay and in vitro chromosomal aberration test, and rest of the assays I will cover in the next part. Before moving to the genetic toxicology assays, we should understand the importance of genetic toxicology assessment. So I have included one case study which is focused on the groundwater metabolites, which is published in the year 2024 in the mutagenesis journal. So what is groundwater metabolites? So those are chemical products formed due to the breakdown of organic and inorganic substances in the groundwater. Those are formed due to the microbial degradation or the hydrolysis or oxidation or photodegradation or the chemical reactions. So why genotoxicity assessment is necessary? Uh, because groundwater metabolites may cause the DNA damage, which leads to mutation and cancer. And regulatory agencies require the safety assessment to ensure the drinking water quality, to identify genotoxic compounds, which helps in pollution control and environmental protection. It also supports the development of bioremediation strategies to neutralize the harmful substances. So, in this study, they have conducted the AIMS in vitro chromosomal aberration assay and the cell gene mutation assay. For that purpose, they have used a groundwater from 18 different contaminants, and the
Dr.
method.
Yogini Soman. Hello. Can you reshare the screen? I can't see anything.
Yeah,
So
sure.
sorry.
I will. Yeah, sure. I will reshare.
Thank you.
Now visible.
Yes, visible.
Yeah, thank you. So methodology
And
was
actually,
sample collection from the groundwater metabolites from 18 different sources, and three battery assays, and data analysis, that is comparison of different assays. So what was the key findings? They have categorized the data in the three different types. So 15 materials, they have found that the AIMS test was negative and the cell gene mutation assay was positive. For two materials, the AIMS is positive and the cell gene mutation was also positive. And for only one material, the AIMS is negative and cell gene mutation is also negative. As So there are discrepancies between the results, so particularly the high false positive results in the cell gene mutation assay, because after getting the positive results in the cell gene mutation test and in the AIMS test, they have conducted in vivo micronucleus test as well for the confirmation, and they found that the chromosomal aberration showed the better predictability, and the AIMS test, it was reliable in overall 18 material. There was a highlight the disparities between the different genotoxicity tests, that is AIMS, in vitro CA and the CGMT, and CGMT which lead false positive results. So it leads to the unnecessary follow-up testing. So you have to conduct the in vivo micronucleus test to confirm the results. And so this study suggests that there is a current regulatory refinement is necessary, and we should support, or we should consider the weight of evidence approach rather than relying solely on the individual test results. So why I have included this case study. There are different battery assays of the AIMS test. First is the bacterial reverse mutation assay. If the AIMS is positive, then you have to conduct one more in vitro and in vivo. So those approach we should consider for the genotoxicity assessment. So now moving forward to the bacterial reverse mutation assay, that is AIMS test, which is developed by the Bruce Ames. So objective of this assay is to evaluate the potential of the test item to induce the point mutations, which revert the mutations present in the tester strains, Salmonella typhimurium or the E. coli, and to restore the functional capabilities of those bacteria to synthesize essential amino acids, histidine or tryptophan. In case of Salmonella, the amino acid is histidine, and in case of E. coli, tryptophan. So you can use different test systems. You can use Salmonella typhimurium 97A or 97 or 1537, or TA9 and TA98, TA100, TA1535, TA102, or E. coli WP2 UVRA, or E. coli WP2 PKM101. These are the ideal models for the genotoxicity assessment due to their mutation detection abilities, the sensitivity to the DNA damage. Also, it gives rapid results within 72 hours, and those are cost effective. So to conduct AIMS as test, you can refer the test guidelines, OECD 471, and for pharma, you can conduct study using OECD 471 and ICH S2R1 guideline. So for the study, you should include test concentrations along with the vehicle and positive controls with and without metabolic activation system. And the study flow is a solubility precipitation test, then the cytotoxicity study, and the main study. So genotypes of tester strains. So in those Salmonella and E. coli, there are different types of mutation. You can see here for 1537, 1535, and T100 or E. coli WP2 UVRA, there is a base pair substitution. From 1537, 97, and 98, there is a frame shift mutation. For E. coli, there is a Occray mutation. So Occray is a type of nonsense mutation. So there is an addition of stop codon there. So now I'm going to explain in detail about the AIMS test. How to conduct the AIMS test. So first, most important part is the solubility of the test item. So before conducting the cytotoxicity assay, first you should perform the formulation method development, and to identify the suitable vehicle for the study. So preferred vehicles are analytical grade water, or the DMSO, or the ethanol or acetone. So you should get a maximum solubility in the test item. Then the second part is a precipitation test. So before conducting actual cytotoxicity study, you should conduct a precipitation assay with the histidine, biotin, and the top agar, and you should see the precipitation in the minimal glucose agar plates. Then you can perform the actual cytotoxicity study to identify the concentration or to select the concentration for the main study. Important things in the AIMS test are the Salmonella or E. coli strains. So well characterized strain, and the metabolic condition with metabolic activation or without metabolic activation. Then the vehicle, as well as the positive controls. Rat liver S9, there is a need of rat liver S9. So why rat liver S9? So rat liver S9 mimics the mammalian metabolism. So S9 fraction contains the cytochrome P40 enzymes, which stimulates the phase 1 metabolism. So it stimulates the oxidation or reduction or hydrolysis. And some compounds, those require enzymatic activation to form DNA reactive metabolites. So we have to include with metabolic activation as well as to mimic the in vivo conditions in with metabolic activation and without metabolic activation. So for the cytotoxicity assay, in the tube you can add your tester strain, then test concentration with metabolic activation. If there is a without metabolic activation, you can add the rat liver S9 there. And there are two methods to conduct the AIMS test. The plate incorporation method, that is you can directly plate entire thing using a top agar which include the histidine, and the another is a pre-incubation method. To increase the exposure of the test item with the test system, you can use a pre-incubation method. So in the diagram, they have showed that there is a rat liver extract and the salmonella strain which require histidine, and the test item which is a possible mutagen, and they have plated entire things in the media with minimal histidine basically, and incubated for 48 to 72 hours. So after the incubation, first is a control plate. Which we get a natural reversions there, so number of colonies are less. And if the compound is mutagenic, we get a higher number of revertants, because his minus is converted into the his plus, so that ability there is a reversion happen, so there are more revertant colonies. So for TA97, TA98, TA100, TA102, if there is a more than twofold increase in the colonies, then when compared to the vehicle control, then the compound is mutagenic. And for 1535 and 1537, if there is more than threefold increase, then the compound is mutagenic. But you should always include vehicle along with the positive controls. So these both should be the validated along with the concentrations. The next assay is the in vitro chromosomal aberration test. So, the objective of in vivo chromosomal aberration test is to evaluate the clastogenic potential of the test item, means structural chromosomal aberrations. So, you can include the test system, human peripheral blood lymphocytes, or CHOK1 cell line. And the test guidelines are OECD 473 for agrochemicals basically. And if a compound is a pharma compound, then you should include OECD 473 along with the ICH S2R1. For the study, you should use validated vehicle and positive controls with and without metabolic activation system. What is the study flow? Again, for the in vitro chromosomal aberration test, first is the solubility test, that is formulation method development. So you should include the compatible vehicle for the study for the test system basically. So preferred vehicles for the in vitro are again, first is a analytical grade water, then the second is a dimethyl sulfoxide, ethanol or acetone. Preferably you should include the soluble vehicle, most soluble vehicle, and maximum possible highest concentration. I forget in the AIMS test that for AIMS test, the maximum possible concentration should be 5 mg per plate. And if there is a cytotoxicity at highest test concentrations, then you can go lower. You should analyze the lower concentrations. For in vitro chromosomal aberration test, you can perform the formulation, that is 202 mg per ml or 200 mg per ml, and from that the working concentration will be the 2 mg per ml. After solubility test, you should conduct the precipitation test and the pH test. For the precipitation test, you should add the test concentrations in the plain RPMI medium. because FBS may interfere with the test item and you get a false results. So if there is a heavy precipitation, then you should reject that concentration and you should go for the lower test concentrations. So slight precipitation or no precipitation doses are acceptable for the cytotoxicity assessment. And again, you should check the pH of the test formulation along with the media. So if there are severe changes in the pH, you can adjust the pH by adding the sodium bicarbonate buffer as well. Then the next part is a cytotoxicity study. So you should incubate the cells with the test system with and without metabolic activation, and you should perform the cytotoxicity assessment. If there is a 50 to 60% cytotoxicity, then you can include that concentration for the main study. If cytotoxicity is less than 50%, then the test concentration is fine. You can include that concentration for the main study as a highest test concentration. If For example, if there are four test concentrations, 2 mg, 1 mg, zero point 0.5 mg, and if the cytotoxicity for 2 and 1 is above the 60%, then you should avoid those concentrations and you should select the concentration which gives the 50 to 60 or below 50% cytotoxicity. And the third one is a third part is a main study. So in the main study, you usually conduct the cytotoxicity assessment as well as visualization of the structural chromosomal aberrations using a microscopic method. So in the in vitro chromosomal aberration test, first the mammalian cells, for example, human lymphocytes or CHO cells, are cultured and those are exposed to the test substance. So treatment is conducted with and without metabolic activation system. So 3 hour with and without, and 24-hour without metabolic activation. Then the cells undergo the normal cell cycle phase. So G1 to S to G2 to M metaphase. If the test item induces a DNA damage during the S phase, that is replication or synthesis phase, it leads to the chromosomal aberration in the later stages. So after the incubation, that is 3 hours or 24 hours. So before basically, so before the to arrest basically the metaphases, the colchicine is added to block the cell division, and the when chromosomes are condensed, and you can see the chromosomal abnormalities such as chromatide breaks, the deletions or the gap or the acyclic fragments, etc., using a microscopic method. You should stain the sample, then you can analyze the sample in the light microscope to detect the structural as well as the numerical aberrations like polyploidy, etc. The frequency of the damaged cells, so those are compared with the negative and the positive controls to determine the clastogenic potential. So you should include the positive or vehicle control or positive controls in the assay. Then only you can analyze. Then only you can determine the clastogenic potential. You can see the figure. I think someone's mic is on. Thank you. You can see in the figure. So there is a chromatid gap here. Then you can see the chromatid gap in another chromosome. Then there is a chromatid break which is larger than a gap. Chromosome break here. And there is a one more chromosome break. So what is the significance of the cell cycle in the in vitro chromosomal aberration test? So DNA damage induced during the S phase synthesis phase, which can result in the chromosomal aberration. Those are visible in the metaphases basically. So cell cycle checkpoints are G1 or S phase or G2 or M phase, which influence the extent of the DNA damage. As cells may repair or undergo the apoptosis if the damage is severe. So this is all about the AIMS test and in vitro chromosomal aberration test. The next is a in vivo chromosomal aberration test. So for in vivo chromosomal aberration test, you can use rat or mice, and The objective is same, to detect the clastogenic potential of the test item. The next is a in vitro chromosomal or in vitro or in vivo micronucleus test. For in vitro micronucleus test, again, you can take a test system, that is human peripheral blood lymphocytes, or else you can use a cell line, a human lymphoblastoid cell line, TK6. And for in vivo, you can use rat or mice as a test system. The objective is to detect the clastogenic and the anogenic potential of the test item. Then the fourth is a cell gene mutation assay to detect the mutagenic potential of the test item. And the other is a comet assay, cystochromatid, transgenic assay, and the in silico approach. So I'm going to cover all the other assays, like in vivo chromosomal aberration, in vivo in vitro micronucleus, and rest of the assays in my second part. So this is all about today. Thank you so much.